Hello, this is Brad Williamson again, and looking at another tool to analyze the data from the AP Biology Artificial Selection Lab. And this particular tool is called JASP. It's a brand new statistical package recommended to me by a colleague, and I'm a little hesitant to recommend it. It's got a lot of power, but it's also very tempting just to let the computer do all the work and not very much, and the student might not be able to do all the thinking because it's so easy. But we'll see how this all works. Let's review the artificial selection lab. We just basically grow up a population of fast plants. We count up the hairs on each individual plant, use those numbers to segregate the population into two. This population over the right is the hairiest population. We grow that all the way to maturity. We pollinate only amongst these, and we collect the seed and plant another generation, count the seeds, the hairs in the second generation, and compare that to the first to see if we've moved the phenotype of trichomes over a little bit, if that population's hairier. And numbers and stats help us make those decisions. So this is where you can get this new statistical package, JASP/stats.org. So let's take a look at how this might work. Now, one of the things that's different about this is you'll still need your spreadsheet where we collected the data. And what you'll need to do in order to get into JASP, because right now it's early in its development, it's in kind of a beta version, and so it doesn't have the power of SPSS, which it's meant to replace, and it doesn't have some of the things that we might want to do, like the ability to actually change the graphs once we've made them. It may be there. I haven't found it yet, but it's still easier to put your data into a spreadsheet and to manipulate it. So here we have our data in a spreadsheet: first generation trichomes, second generation. Basically, all we want to do here is we want to save as, and save this spreadsheet as a comma separated value, a .csv. And we save it as trichomes, and I'll save it on the desktop, and I'll replace the one I already have there, continue, and now I'm ready to go work and do the descriptive stats, or do the display of the descriptive stats in JASP. So here we go. I have already imported it, but to do that, all I really have to do is hit File and Open, and it'll just automatically open it, which is kind of cool. Now, you see it's actually trying to do that already. Now, let's go ahead and do descriptive stats. First generation. Boom. There it is. This is what I mean by it's so easy. It just did all my descriptive stats. Mean, standard deviation, minimum, maximum. It even drew a histogram and even did a plot of a best fit line to see how it might look like a normal curve. I can do the second. Boom, it's there too. And now I can actually, and on top of that, if I want to put it into a report, I just copy it. It's copied on the clipboard. I bring up Word. Oh, I've already got one there. I'll show you how easy this is. I'll drop it here, paste it. Not enough memory. I'm running out of memory space. But it actually pastes very easily into Word. I'm going to just shut Word down. It's taking up too much space. Okay. Now, that's not everything we want to do. We also want to do hypothesis testing. And right now, looking at these two plots, it's kind of hard to tell. It looks like this one is shifted over, but it's not enough to make an argument, nor have we created any particular bar graphs or such. So let's consider that the problem here is that this particular data, the form of the data table, won't work. So here's what we have to do. I'm going to add a column. This is part of the SPSS and other statistical packages like R. What we have to do is we got to change things a little bit. We're going to put first generation right here. And I'll make that a little wider. All right. We'll do that again. In fact, what we're going to do is we're going to copy that down, and we're going to fill that all the way down. and this is going to be our generation variable, and it's our grouping variable, and this is going to be our trichome variable. Now the problem is there's only right now the first generation. We got to get this second generation down here. So I'm going to highlight that. I'm going to copy. Oh, what did I do? Make sure. Let's just take that out. Command X. I'm going to come down here and put it right here. Come on, back here. Oh, let's see if I can get it reversed. There we go. I'm going to put it right here. So I've moved that, and this is my second generation. Do another one. Now I'm going to just copy that. And now I've created a different format, the kind of format you probably would never use with your students, but what you need for the statistical package. And all we have to do is, again, do a save as and save it as a comma separated values file. It'll come up here. Make sure that right here you say CSV and we'll put a different name. Do a save, and we now have that. It's ready to go into JASP. Here it is. You can see it looks a little different this time, but this time we can do a t-test as a hypothesis, null hypothesis. And the null hypothesis is these two generations are not different. They simply essentially have the same distribution of plant hairs. So I'm going to do an independent samples t-test. It's already over here, ready to go. We're going to put trichomes under the dependent. We're going to put generation under the grouping variables. Now, I didn't actually load that up. That was a previous one, and this is one of the problems. If you don't have that actually typed in the way you want it, it's not going to work. Right now, I used an abbreviation earlier, but check how quick that was. T values. I've got all these things clicked. So I've got a lot of things like 95% confidence. I've got first gen, second gen, mean, standard error. I got all this stuff just showed up with just one click. And check this out. Descriptive plot. Boom. Look how beautiful that is. First gen, second gen. This is an appropriate graph. It's got your trichomes over here. All of this highlights and copies just like that. Ready to go into a Word lab report or onto a poster. And notice up here, all of these stats up here, ANOVA, regression, frequencies is chi-square. These are basically the common things that you would use in an AP biology classroom. And again, it's because it's new, we don't have an ability to actually modify this graph except by modifying it at the beginning. It does have Bayesian statistics, which is actually its actual goal in making this particular software package if you want to explore, but you don't have to. You can just stay right up here in this area right here. Regression analysis right there, very clear. Contingency tables. I'll do a video on how to build those because they're a little different also on the mitosis lab. But I think this JASP is something people ought to give a shot at, and the AP biology community. Hope to hear from you if you do. Catch you later.